Good evening, everyone. Welcome to the fourth session of our PH2O webinar series, Cytometrics and Rapid Microbial Methods in Pharma Water and Bioprocessing. My name is Shivani, and I'm delighted to have you all with us today. At CN Water, we are committed to advancing digital and microbial intelligence in clean utilities, and today's session reflects that very mission. I extend a warm welcome to our distinguished panelists who will be sharing their valuable insights with us on today's topic. So, first we have Dr. Jürgen Illerhaus, who joins us from Switzerland. With a doctorate in biophysics, he has over 20 years of experience in validation, aseptic processing, and instrumentation design. His career spans the development and commercialization of advanced analytical tools for regulated pharma environments. And at BWT Pharma, he focuses on building market visibility and adoption of core products and emerging technologies like online bioburden monitoring. So, Dr. Jürgen, we welcome you.
Thank you very much.
And next, I would like to introduce Dr. Sanjeet Singh Lamba. Dr. Lamba has had an illustrious journey of over 36 years and is a leading voice in the Indian pharmaceutical industry. He has led large global teams in organizations like Merck, Pfizer, Ranbaxy, Dabur, Gland, and Lupin. He also held the position of a managing director at the Japanese company ISAG. He is a founding member and past chair of PDA India chapter and has served as chairman of the Technical and Supply Chain Committee at OPPI. Presently, as founder of Trillium Consulting, Dr. Lamba serves as an independent director, coach, and advisor to several leading pharma organizations. Dr. Sanjeev, welcome.
Thank you very much.
We are so honored to have you both on the panel today. So, in today's discussion, we will explore two key drivers of innovation in microbial testing, that is RMM and cytometrics. The panelists will share a global point of view, presenting their analysis of key trends and advancements in these areas. As for our listeners, we encourage you to post your thoughts and questions in the chat, and we'll take them up during the audience Q&A segment at the very end of the webinar. So let us now invite our speakers to begin the discussion. First, I request Dr. Jürgen to shed some light on key technology, regulatory trends, and the methodological evolution of pharma water globally. So, Dr. Jürgen, the floor is all yours.
So,
Yes,
Shivani, thank you very much for inviting me to this very interesting and exciting event to present flow cytometry as a potential candidate for online bioburden monitoring in the future to the Indian market. It is a big pleasure for me to introduce this technology because we are investing a lot of time, a lot of effort, and are in contact with customers and authorities to find the best possible solution for bioburden monitoring in water applications. Before I want to go into more details about this technology and to explain to you how it is working, I would like to describe a little bit the motivation of BWT for the introduction of such a bioburden monitoring system. Maybe all of you know BWT from our water purification equipment. BWT is quite famous worldwide in producing water purification equipment, hot WFI productions with distillation, membrane-based systems, and hot steam production. So why are we dealing with such an idea of in-process control for bioburden monitoring? Because this in the past was the main focus was the end control, the final control at the point of use to guarantee that the water at the end, where the user is needing the water, that the water quality meets the regulatory requirements. For us, one of the main drivers was clearly the in-process control. Bioburden monitoring is basically the last parameter that is important to be monitored, and this is the last which is done manually today. And manual work and having a result just a week after we take a sample this is not something we can use for IPC. So we need to have a reliable measurement, we need to have a fast result, and we need something that is able to be automated. On the other side, we didn't forget about the QC at the end of the day, because QC, this is a little bit a different story. There we talk a lot about regulatory requirements, about compliance, about compendial methods and something like that. For IPC, we can use basically whatever technology which is available in the market. So what we know since decades, the limitation of HPC. HPC is not usable from our perspective for in-process control. It takes too long. And the main disadvantage of the HPC, heterotrophic plate count, is that it really monitors only about 1% of the total bioburden in a water system. And we, as a manufacturer of the water systems, we want to know the total bioburden and not just the colony forming units. The majority of the microorganisms in our water systems are the so-called VBNC organisms, viable but not culturable. This we also want to know what's going on in our water system with these organisms. And in 2022 or 2023, there was an interesting publication at the university in Germany where a team applied stress to microorganisms. And after a short period of time, these microorganisms were not visible on a plate count anymore. They didn't grow on the plate anymore. But if you release this stress after a while, even after 10 months of application of this stress, it takes a while, but suddenly the microorganisms start to grow again. Throughout all this period, the total number of microorganisms did not really decrease, but they were not visible on a plate count anymore. So this team came to the statement, currently the viable but not culturable organisms or state is an underappreciated niche for pathogenic bacteria, which provides a continuous source of recurring infections and transmissions. So this was also a point for us. We have been looking for a technology which shows the total bioburden and not just colony forming units. And then, last but not least, the Annex 1, most of you probably know that, has been released in August 2022 and is legally binding since August 23. And there it's clearly the demand for a proper in-process control, for a contamination control strategy. And this means, from our perspective, from our point of view, we need to understand each and every step of the water generation systems. We want to monitor this. We want to know what's going on to take corrective actions as soon as possible. Years of investigation. We really invested a lot of time starting in 2017 to understand what technologies are available in the market. We have been testing whatever was available, whatever had a chance to be usable as an online instrument. And for us, we came to the conclusion that the best technology in the market, the technology with the best specificity, which is also proven technology in many applications. You find flow cytometry in laboratories, in chemical industry, in pharmaceutical industry, in food and beverage industry, wherever microorganisms need to be counted or wherever the bioburden in a sample needs to be defined. It is a proven technology. It counts intact cells and it basically has no background due to the measurement procedure. And the beauty was we found an instrument, you see it here on the left side, we call it Aquasense MB. It origins from an instrument that has been already in the market for some while for drinking water analysis. It's a small, very compact unit, very robust, and it is really accepted in many applications already, especially in the Swiss drinking water monitoring. This is already accepted by the authority to use this instrument for bioburden monitoring. And the beauty was it was basically ready to develop it for pharmaceutical applications where we are talking about very low numbers of microorganisms in contrast to very high numbers that we have in surface water or drinking water applications. And on the right side, you see how it is installed to a water generation system. Now, to understand how these technologies, how this can be used for controlling for in-process and later on for quality control, I first want to introduce the instrument and the technology, how it is working. For this, you see here a short video which describes the process when an instrument is installed online to a water generation system. So what happens during that? The system, as I said, directly connected to the process, takes a sample about every half an hour or longer. It mixes this sample with a staining solution. And that's very important, this step. It's a staining that is already known since many years. We are using two different stains, propydium iodide and cyber green, and they do not interact with any organic or inorganic particles. They are DNA specific. They only stain DNA molecules. And only the cyber green is able to enter an intact cell. The propydium iodide cannot pass an intact cell membrane. Only damaged cells or fragments can be stained with the propydium iodide as well. So based on this staining, we have a very strong specificity for living organisms. After incubation and mixing, about 15 minutes or so, the sample is now pumped through a very, very thin capillary and is illuminated with a laser beam. The laser beam generates three different signals. One is a stray light, the blue line, and two fluorescence lights from the two staining from the two dyes that we are using for DNA staining. And each and every event which passes this very thin capillary, and they pass it one after the other, generates a signal based of these three different signals. On the right side, you see here the so-called dot plot. Each event generates one dot in this dot plot, and the dot plot is on the x-axis the green fluorescence and on the y-axis the red fluorescence. And now the system is running the sample and counts each and every event. And what you see here in green, these are the living cells. And this we can say because we made a primary validation, and during this primary validation, we created a so-called gate, which is a line which separates the intact cells from the damaged cells or from the dead cells. And that's a very reliable, robust technology. It runs fully automatically. You can run up to 1,000 measurements with this instrument without any user intervention. After these 1,000 measurements, you just have to replace the liquids inside. It's a closed cartridge. You do not even get in touch with the liquids, and then you can run another 1,000 measurements. And this is basically how a so-called dot plot looks like. You have this pattern. X-axis is the green fluorescence, y-axis is the red fluorescence. And as I said, each and every event generates one dot. And what you find here is the living cells below this so-called gate. You have some dead or damaged cells, particles outside, electronic noise, something like that. So you can clearly differentiate between living cells and anything else. The beauty is here you find about 95 to 99% of all living organisms in the water sample. And for the let's say the power of this instrument, just a small number. In the Zurich drinking water, we have 1.5 million microorganisms in a sample in 1 milliliter. We are measuring 100 microliter sample, and we can count these numbers of microorganisms, each microorganisms individually. With this, we get signals. What you see here for specificity, for example, I show you these data. You see that different microorganisms appear in different areas of this so-called dot plot. Ralstonia piquetti is at a different place than Pseudomonas aeruginosa. You see PTFE beads, for example, iron oxides give absolutely no signal in this gate that we are where we are measuring. All the particles outside and the electronic noise is far away from our measurement area. So This technology, from our perspective, is the strongest, the most potential technology for really counting microorganisms and counting living microorganisms. And that's what we have been looking for. What is very important to understand, ICC, the intact cell count, has absolutely nothing to do with colony forming units. And it's by orders of magnitudes higher values than you have for colony forming units. And this needs to be considered for all future aspects of validation of using this technology. To be honest, this is also the hurdle now to introduce it for quality control, but we are on the way with some key customers worldwide to do the full validation of this technology to be able to use it for quality control aspects. Now, to explain and to show you how a measurement, a real measurement would look like, here is a pattern of or a graph of a measurement, an online system connected to a WFI system after ultrafiltration for a time frame here of 43 days. You see at the beginning until, let's say, day 26, a very stable signal. This is basically the control level or the level where a well-maintained system can work. We have numbers that are below 50, a lot of zero values, and but very low values. And you see after a while, okay, the numbers are increasing. There's obviously something changing in the system. Now, how can we explain that? How can we explain this change? To explain that, we need to understand how our microorganisms live in the water system. Usually, microorganisms like to build conglomerates. So you have to consider individual cells, individual culturable cells, individual non-culturable cells, the VB&C cells, but also conglomerates of cells which may be attached to a particle, a rubber particle, a rouging particle. Or you have a biofilm fragment, a small biofilm fragment, something like that. So all these events could be counted by the Aquasense MB, by this technology. So what we expect, and this is still a hypothesis, to say this background signal, this is usually the individual cells. There we are talking about 1, 2, or 3 cells in one sample, since we just have a small sample volume. But from time to time, or you see higher values. You see values up to 300, 500, and this most probably are smaller conglomerates of cells, a particle with attached microorganisms. And you have very high numbers. They can even go up to thousands of cells in one sample, but this can, from our perspective, only be explained by biofilm fragments. So you get already more information about your processes than just the number of microorganisms. You get some ideas about what's happening with biofilm formation, something like that. If you would now see this test here, and you would measure with HPC on a colony forming unit base, you would see individual culturable cells here just one by one. If there's one individual cell, you would see one colony. If there's a particle or conglomerate with culturable cells in it, you would just see one event, one colony on the plate. Biofilm fragments, you would also just see one colony. With flow cytometry, you would see the number of each individual cell in this volume that you have tested. In this example here, you have one individual culturable, you have one conglomerate with 10 microorganisms, you have a biofilm fragment with 18 microorganisms, and you have one individual VB&C organism. That is what you basically see here in this measurement. And as I said, so far it is a hypothesis. Now there was a study published at a congress in Switzerland, end of 22, I think, where a team has compared different methods They compared HPC with solid-state cytometry and this with flow cytometry with the Aquasense MB. Solid-state cytometry is you filter a sample, you stain the sample with fluorescing dyes. In this case, it was also DNA-specific staining. And then you analyze it under a specific reader to find events on this membrane. And you see from the numbers here, sample 1, 0 HPC, 0 colony forming units, 46 events on this solid-state cytometry, and 1,403 with Aquasense MB, and so on for the other samples. Now let's look into this test a little bit more in detail. This was a picture of a plate done in this test, 33 colony forming units. Flow cytometry has given 115 events for living cells, and the Aquasense MB has given 2,084. And if we look a little bit more in detail on this picture of solid-state cytometry, you see each event consists of many different individual cells. So flow cytometry shows each and every cell in this sample. So that's the beauty of this technology. What we did now, we made a primary validation for this instrument. We partnered with universities in Switzerland, in Holland, for example, and we were able to show that the Aquasense MB was passing all individual tests for the primary validation. So the primary validation was successful. Now, if you look into validation studies, just to show you an example how data can be analyzed, this is a half year's test at a customer. You see the green bar is showing basically the background of microorganisms in this sample. You see individual high values, and at the end, suddenly there is obviously something changing. Interesting here, there was no significant change in HPC on the plate. We have not seen this effect on plate count. Now, to analyze these data, we can just apply standard methods that are used for environmental monitoring. It is so-called trending, quantitative trending rules. You can describe different sets of trending rules. It's nicely described in the book from Mr. Montgomery, Statistical Quality Control. There are nice examples how this method can be used. And if you would apply these trending rules that we think are applicable here, you see at this point, all trending rules are broken. So here we have to say at that point, the system is out of control latest at this point. Now to go a step further, to have more criteria to describe the system, we can do a percentile calculation. Percentile calculation just gives you the number of how often individual values appear in a measurement sequence. So you see here the majority of the measurements have been zeros. It's about 80.4%. The tens, 20, and so on were lower numbers, and we have seen that basically 100% of all values in this sequence were 100 or below. Now, when we just analyze the data until the point where we say the system is out of control based on the trending rules, you see that suddenly 90% of the values are zeros, 8.3 are tens, and so on. So 99.9% of all values were 30 or below. Now you can generate, based on these data, rules. You can apply the 99.9 or the 99.99 percentile, and you can use this as action or alarm levels. So just by doing the data analysis with these two methods, quantitative trending rules or percentile calculation, you can also already implement your limits for your systems. Now this means, what do we get? We have a control level, And this complies fully with the contamination control strategy. You want to control your system on a specific level. You have an out of expectation, it's an individual value, probably one biofilm fragment. And you have here, obviously, the system is out of control. So now it's time to do something. Throughout all this period here, the system was not out of spec by using the heterotrophic plate count. So the next steps for a validation, you need to do some internal testing, you do some in-house isolates, you have to redo some parts of the primary validation, and you do a non-inferiority study. That's what we are now doing together with our customers, worldwide companies in United States, in France, in Nordic countries. So we are on the way to do the full validation together with them. We can offer PQ documents and everything. So our ideal picture for an AquaSense MB, for the implementation of this new technology is one part is you're able to do an excellent in-process control, monitoring each and every manufacturing step for your water systems. You see immediately if there's some changes, if the system is running out of control. The second application is the QC application, and again, from the point of view of the system manufacturer, not from the point of view of the point of use. This is a different story. This is not the core competence of BWT. There you can apply routines, for example, by using offline measurement, sampling, using a sample manager with the AquaSense MB and measuring the sample. The typical installation we have now in Switzerland, in Germany, in the United States is what you see on the right side, that you combine the output of the generation system and the return line from the distribution system to one AquaSense MB. You monitor both measurement points, and then you can describe, and then you have the proof that your water system and the water distribution system is under control. That's what I wanted to show you in due to the time, we cannot go much deeper into the details, but our team in India is very open for your questions and supporting you if there are more questions about that, more detailed information needed, and I'm also available for that. So thank you for your attention.
Thank you so much, Dr. Jürgen. There were so many great takeaways from that. And I understand how adopting such technologies will be so crucial to ensure compliance and product safety in the near future. It is important to find the best way to optimize time along with accuracy as well as data handling. So, such technologies will help us achieve that. So,
Yeah.
now let us shift gears a bit and invite Dr. Sanjeet, who brings deep experience from the Indian industry, regulatory shifts, and digital transformation trends in India. So, over to you, Dr. Sanjeet.
Thank you. So this was a very interesting uh, kind of work which has been done and shown by Dr. Yogan, and I really enjoyed some of the very finer points of validation data. Let me just share my screen, please. So thank you very much. And in fact, this is one area which is very close to my heart. And then I think a lot of my questions, which I had in mind, Dr. Yogan, you could explain it so brilliantly in a very short time. And I would like to, first of all, thank the CN Water Systems and BWT for inviting me here today. And I would like to share my thoughts from the industry perspective and what a customer or in industry perspective is to look at rapid microbiology monitoring using flow cytometry in the water system. So I would say, first of all, what I would think which comes to my mind is this is not an automation. So it's a really a transformative technology for rapidly monitoring through a microbiological method in water systems. And you are having a significant advantage over the efficacy about the time, the speed, the sensitivity, and ability to provide a real time data for ensuring that the decision making is helpful. So I would say this is connected operations, which are actually tailored to provide solutions to the pharmaceutical sector, looking at the regulatory and operational needs of the organizations. So flow cytometry, as you could see in the video, could really give us an online monitoring and see what is going on in the system and why we are either in the state of control or we are going out of control. So these are very effective fluorescent dyes based distinguishing procedure from non-biological particles, which Dr. Yugan explained. And I think we are able to see both total and intact cell counts in this area, which is really the interest for the organizations. So I think from the industry perspective, what we are looking at, this can provide speed, sensitivity, the resolution, what we are looking at, and of course, it is an online system for continuous monitoring. And I would say this is one area which can satisfy the needs of the regulatory agencies for not only real-time data collection and analytics, but also an end-to-end kind of a digitalization where the data is stored, which further enhances the data integrity and the compliance what the industry is looking for. So the system is very flexible and also very provides you predictive kind of technology to look at early warning signs where the contamination we are able to determine that there is a contamination or a regrowth happening in the system. So it helps really the water quality management in a larger way. Uh, Past we have seen there were some issues about the limitations and of the rapid microbial monitoring systems for water systems, and these were related to non-specificity. But I'm happy to see that systems of the flow cytometry, it provides total and intact cell counts, which help a lot in real monitoring. And also there is, as Dr. Yugan said, there's no background interference which is happening from whether it is debris or particulates or from the rust particles and iron oxide, which I could see in his presentation. These are some of the concerns in the past couple of years, which organizations have shared in some of the surveys which were conducted on rapid microbiological monitoring. Another limitation earlier was, there is a expertise sometimes which is lacking in some of the systems. In many cases, you don't get the advanced metrics or the computational techniques for ensuring that your data is validated. But in this case, I think in one of the systems for BWT, which I could see, there is specialized expertise available with people where a lot of data is available as far as the validation is concerned. And I would say, as I said, this is really industry transformation which is happening. And what a regulator is looking at from last many, many years is a process analytical technology. And this particular technology of flow cytometry really encourages rapid microbiological monitoring for parametric release for an online system, because we are very, very concerned sometimes that you are looking at batch manufacturing and you're not sure about what is my quality of water which I'm going to use. And I'm going to rely on the data which is going to be released much later after my product is manufactured. So this is really a very predictive online continuous process monitoring, which I would say is really a revolution in the water management. And also this aligns with the quality by design principles, which enhances your operational agility and also competitive positioning. So I think from the regulatory perspective, there has been a lot of work in the background which has been done. As we know, today the USP standards, they require that the rapid microbiological monitoring should demonstrate non-inferiority to compendial methods like USP 71 for sterility testing or for water management or for other areas. And a lot of other areas are also being researched today how it can help in online monitoring. And things like, if you see the USP 1223 and 1225 for alternate microbiological monitoring methods, there are systems and details and guidances available for this. Also, organizations and regulatory agencies like Health Canada, it permits rapid microbial methods only if the validation proves equivalence to established techniques. And in this case, the data which we saw in Dr. Jurgen's presentation shows very clearly that it's equivalent, or it can give you more data than what is expected. One of the very interesting areas is, which is happening from the last 4 or 5 years, is agencies like US FDA, they launched a special program about data modernization action plan to look at how it can really be designed for organizations where the organizations are using more digital and data driven online monitoring systems, and how this action plan can work and help these organizations. So a lot of work is being done in the background, and you will see more and more rapid methods uh, being implemented in future. And of course, we we are looking forward in India to implement some of these systems, because some of the facilities in India are really state of art, and we need to do much more than what we are doing to help us in agility, help us in transparency, help us in compliance, and also data integrity assurance. And this is a really a step forward in that direction. So, of course, when you look at from on-ground challenges point of view, there has been a lot of concerns which have been shown in some of the surveys conducted by some of the organizations, which include technical limitations of delayed microbial detection, especially for low bioburden samples, very low bioburden samples, or stressed samples, which Dr. Yogan spoke about. And I think it's very important for the process to be very specific. And also, some of the regulators, they have shared that validating such systems requires substantial investment and expertise. And in my opinion, I think this is an ongoing process which is coming up. And as we could see some of the case studies where a system as small as a box is having a laser system online for detection of your uh, contamination. So it's not only an online monitoring. It's if you look at Annex One, you can say that this is one uh, transformative step to ensure compliance, and especially contamination control strategy, and how your water systems can really adapt to the new guidance requirement of contamination control strategy using an online RMM system. And I think this is a great a step forward in that direction. Of course, one of the areas which some of the organizations have shared is that they are using legacy systems, and every organization is using a water system. They're manufacturing a pharma product, but some of the organizations have shared that we want to implement a new rapid microbial method systems. What to do with the legacy systems, and how the systems can be retrofitted on the existing system. So this is one of the concerns some of the organizations have shared. I would like to say that, as I said, this is a journey which is a step towards Pharma 4.0, because it's a really transformative step in a real-time data control and a parametric release, and ensuring that there is a continuous quality assurance. Because Pharma 4.0 is a journey. It's not an on-off system that you are there or not there, but it is a step toward this direction. And I think a rapid microbial monitoring system ensures faster, more accurate contamination detection, especially for water systems. And of course, there are a number of other applications which are on the way, and you will see more and more happening in future. But if you see from the regulatory perspective, a lot of modernization efforts are on the way. As we have spoken about process analytical technology, I know for many, many years, We have a guidance, but the some of the real implementations are delayed a lot. But I believe that especially, I would say, especially from regulatory compliance perspective, there is a larger issue of ensuring a data integrity in a microbiology lab today. You will see number of warning letters and also 483s, especially on looking at how the microbiologists and the overall system in microbiology lab, as well as areas like water systems, how do you assure that your data is really intact? And what is your data governance system for ensuring the data is integral in microbiology labs? Because there's a more of manual systems by HPC monitoring, which Dr. Yugan explained, which are more traditional mass methods. And the colony counting systems and other areas have a lot of areas, gray areas, where there are chances of data integrity failures. And I think this is really a great effort for organizations who want to strengthen their data integrity assurance and governance mechanisms. I would say from the policy changes, you will see a number of things happening both in industry as well as in the agencies, as far as the gaps which are existing, and the gaps are related with validation, because the systems are complex than the existing systems for, and I think it's more expensive for smaller manufacturers because it's difficult to implement, and they are looking for a cost-effective methods to ensure water quality. So, how can these systems become cost-effective in future is something which is we need to look at, apart from not only the capital expenditure but also the validation requirements, which are more complex. But the way I could see with the presentation from Dr. Yogan, the validation documentation and assurance is already provided by BWT, which is great. And also, I would say we need to really look at how we will see more artificial intelligence um, driven cytometry being implemented more and more into rapid microbial monitoring, and how we can ensure that there is a global consistency in accepting such results, and how do you really evaluate your action and alert levels. So probably I would say the future, uh, the organizations are looking for three key developments which will dominate the pharmaceutical quality policies in various organizations and as well as regulatory agencies, is to really look at real-time release for products as well as materials and input materials like water, and how do we ensure that there's a continuous manufacturing. And for organizations who are already and early adopters of continuous manufacturing, this is really a step which will integrate the RMM into their core components of quality by design principles. And also from you will see using the rapid microbiological monitoring, and organizations can really strengthen their contamination control program by using a dynamic risk assessment models more and more to get an assurance that their systems in control. So you have seen more into already with Dr. Yugan's presentation how it can be used in the water systems. What I would say, we are looking for water quality to remain in a state of control, and make sure that we have assurance of regulatory compliance, because there are a number of questions which are getting answered with the rapid microbial monitoring system. And it's a complete shift which is happening, and helping us to create multiple trending uh, to ensure that there's a continuous quality assurance, there's a continuous monitoring, there is a data driven decision making process, and also there is a focus by the organization on more preventive areas like minimizing the biofilm formation. How do we ensure that our sanitization systems are designed in such a way that we can predict when the system is getting into a contamination, so we can start our preventive maintenance and sanitization schedules in line with that. I think this is a lot of benefit which will be there for the industry. And also, There are a number of concerns regarding sampling practices, how the sampling is conducted in the industry, person to person, individual to individual. There are a number of areas depends upon the skill level, and this is an online system based on a validated data and the risk assessment, which is much more effective in assuring data integrity and compliance. So it will ensure more adherence to standards, more adherence to our monitoring programs. So I would say, in summary, that this flow cytometry, it really represents a rapid, robust, highly informative system to approach microbiological monitoring in water system to ensure we are in line with the Pharma 4.0. We are really looking at improved water quality management, early detection of contamination events, and more effective process control. So, well, really, I would say this is not a technology which would replace the traditional methods for pathogen identification, but probably I think this would be a very, very essential tool now and also in future in most of the modern facilities requiring water utilities. And how do we assure from the regulatory compliance and data integrity compliance with an online data governance management? This is a really step forward in this direction. We are really yet to see the implementations here in India, but I'm saying that looking at this kind of a presentation and data from Dr. Yugan today, and organizations are really evaluating today, it's a really a wait and watch situation of implementing such systems. But this is a really good step to consider for organizations. Thank you very much for your patient listening.
Thank you so much, sir, for analyzing some very important facets of RMM and cytometric techniques. You have helped us get a broader picture on how they have transformed the industry and the various policy frameworks and on ground challenges that need to be understood thoroughly in order to harness the full potential of these techniques. So, thank you very much. And now the floor is open for the audience. So, feel free to drop your questions in the chat and we will take them up with our speakers. Okay, so a lot of questions pouring in. The first question is for Dr. Jürgen. So you took us through your new technology. Could you summarize, give us a side by side on how this new technology is superior to our conventional method? And adding on to that, how popular is this technology already on a global scale?
Yeah. So the way how we are using today this technology with our customers, with our big customers, is basically doing parallel monitoring to the conventional plate counting. Because, as I said, the technology, it's not new, but in this application, it's something completely new. So people need to understand first what numbers are we measuring. They are totally different from what you're expecting on the plate count. So even in a super clean water system, you might have ICC counts of about 50 per milliliter. This is 5,000 per 100 milliliter. So you have to deal with completely new numbers. This is the first thing. Important is trending. Important is having a lot of data, is seeing how does my system change over time. And that's basically the way how customers are using it today. And based on this much higher sensitivity of this flow cytometry, they are able to avoid any uh, any problematic development based on the heterotrophic plate count. They will see the customers, the users, see changes in the water system much long before you see something on a plate. So you avoid problems. You can control your sanitization processes. You can schedule your sanitization process, and that's basically how our customers are using it today. In parallel, some of them are already doing the full validation for quality control. Another topic, for example, is our service team. Our service team is using the Aquasense MB to identify the source of problems as soon as a customer has an unexpected contamination in his water systems. It can come from a point of use, it can come from one of the components in a water generation system. So this is the application, how we are using this instrument on a daily basis. And we are able to identify the source of a problem within a few hours, within a very short period of time. We are able to take corrective actions immediately and prove the effectiveness of this uh, at the same day. And now you can imagine how long it would take if you only use heterotrophic plate count. It takes you weeks until you have the proof that the water is clean. With an Aquasense MB, you have the proof at the same day. And that's basically how we are using it. And from the customer segments, I have to say, it's basically all big pharmaceutical industries, all big companies are now in testing phase, some of them already in validation phase. Everybody is aware that new technologies have serious benefits or bear serious benefits for their systems. And so the attention increased in the market significantly, and especially after releasing the Annex 1, more and more customers are approaching us for support on that topic.
I would like to extend this question to Dr. Sanjeet. So, Dr. Jurgen gave us an idea on the global level. So, can you give us an idea about the Indian market? So, what are the hurdles being faced in the Indian market? Is it cost, infrastructure, or the mindset in general?
So I think India, as you know, is a very vast country. There are close to 8,000 plus companies operating in India, from a very small to medium to large enterprises. And most of the large enterprises today, they are having a state of art facilities, including some of the medium scale organizations, of implementing facilities containing both purified water as well as water for injection. And they've been really in a state of control. More and more, whenever we're looking at expansions, more and more facilities are coming up in India because there's a lot of expansion happening here. So I think the challenges are many. One, of course, is if you look at a pharmaceutical company in India, people are looking at more cost effective solutions, of course, value driven, but more cost effective solutions. Here, I would say that it's an too early to comment right now, because I think the system may have some more investment in the beginning, but probably the value it will generate would be huge, because then you have a real time continuous quality assurance getting built into the system. So, of course, cost is one area, infrastructure, and how much a company is ready to invest in predictive technologies and close to real time continuous manufacturing is something which is yet to be seen. Another thing, it's a really mindset, I would say, of an organization. Sometimes they are not very concerned about the only the investment, but the mindset is like, I would, whenever there is a pharmacopoeial method of HPC, I would continue to use that. Why I should drift from those practices and policies, because they're time tested and the agencies don't have issues regarding that. So they would like to stick, and so the status quo needs to be maintained in such situations. So change is considered very difficult, and I would say this is a really big change, from moving from a traditional system to an online monitoring system. But I would say a change is always looks difficult in the beginning, and later on, when real benefit starts, everybody says why we were late, why we did not evaluate and implement this earlier. So it's like a catch 22 situation. Who will start, who will bell the cat first, where I'll get the data, which organization would start. I would say 20 years back, when I came out with the X-ray diffraction system for rapid inspection of powder-filled vials in one company in India, and I worked with US FDA for early implementation in India. And I went through a lot of resistance initially, but today you will see more than 2,000 machines operating in India across the world on X-ray inspection systems for aseptically filled powder vials, where there was no technique available earlier. So initially, I would say we had to do a lot of work, but today it's really a technology available to you. And same is the case, I would say, not only in the water systems, but also in the sterility testing, but also in environmental monitoring. There's a lot of work which needs to be done, but water is a very, very important resource for an organization. So I think it's a combination effect, Shivani, which is a combination of a mindset, initial investment, and how they're going to really take care of the legacy systems, how the new data generation will get integrated with their legacy systems. These are the questions, and along with the validation efforts which are required for this. But I think if you have a good support mechanism available in India to handhold the organizations, you will find some early adopters, people like me sitting across to really implement a new technology. You will find people who want to make sure that we have an early benefit in these areas. So I think India also is very, very progressive, and there are very progressive organizations in India who would really look forward for implementation in this.
Right, I completely agree with you. More like you said, it's a wait and watch situation. And once
Yes.
some people take this up, I'm sure it will be a domino effect.
Yes, absolutely.
That's what we see in Europe and United States already. Still slow, but now it's speeding up significantly.
Absolutely.
Absolutely.
Maybe there's one other aspect we could mention here. In the pharmacopoeia worldwide and other documentations, it's written since years that the
Yes.
limitation of HPC, of heterotrophic plate count, is that you don't see 99% of the living organisms in the water. This is known since decades. And now,
Right.
since there are technologies with which you can monitor this part, now the authorities approach us. Even I have been in Germany at the authority yesterday to present this new technologies because they say now there is a tool to monitor
Yes,
this. Now please let's use it. Let's do that.
exactly.
And
Yes,
that's an important point. It will not change from today to tomorrow, but I believe that in the near future there will be demands from the authority to have a look on this part of the quality of the water.
exactly.
Yes, very true. Okay, so speakers, we are running a little bit short on time, so do you mind if we take just a couple more questions and then we'll conclude?
Sure.
Yeah. So my next question is for Dr. Yogan. Is there any temperature limitation for this RMM technology?
A temperature range is the staining process needs to be performed at slightly below 40 degrees C, and that's when you have an ambient system, easy, it will be heated up to this temperature. And if you have a hot water system, you need a small cooler in front of the Aquasense MB to cool down the sample to about 40 degree. This is a very simple installation, very easy to do. The system is fully sanitizable, hot water and ozone sanitizable. You can run hot water through the system, but only when you want to do a measurement, you should cool down to about 40 degree for doing the measurement. But it's technically very easy.
Now we'll take the last question, which is to Dr. Sanjeet. What measures can be taken to integrate such technology into our legacy water systems without disrupting how they're presently functioning?
Probably, if you look at, if you're looking at a legacy system today, one of the important areas where this system can be used initially is to look at a situation how your water generation system is working, and also look at what is at the return level, what is the level of contamination which you are observing. And this study probably you need to do over a period of time to look at whether your existing systems and your offline testing method, which is there, which is a colony counting method, is it aligned with the expectations today. And I think we need to generate that data first of all before implementing directly an online system, where in offline mode you are able to detect uh, any variations, because in general in companies we have a system of sanitization, preventive maintenance, and sampling. How your sampling technique or a statistical sampling is aligned with a real count of the contamination in your system. So I think first of all, a lot of groundwork needs to be done in looking at what is my actual data rather than what I see on an HPC, because as the USP itself says, that level of detection today is very low. So now, once we have that kind of a trend available, even if it is a legacy system, I think we need to look at organizations first which are facing issues about where is the issue today in my generation plant or in my distribution loops. Where is the contamination? What is the root cause of this contamination? And I think this flow cytometry can really help in diagnosing where the issues are. First of all, I think this is what we need to do more and more, because right now what we are doing, a sampling technique, what we are following, it takes time and does not give us data about the stressed microorganisms which are there in the system. You are doing sanitization, you are doing a lot of cleaning, and how it impacts the real count. This background data, this baseline information can really help you to move from an offline system to an online system. And I think if the validation support is really good, and we can really move from an offline data to an online mode, and can see how we can actually take first a sample from the system and can do parallelly both HPC as well as my online monitoring, and see what is the kind of a trend which is there. Is there a kind of a alignment in this case, or we still have issues? Because I think more and more needs to be done in the planning part, in the validation part, in ensuring that this system complies to my USP method for microbiological monitoring of water systems. And I think we need to, and I would say more from the mindset point of view, we really need to have a consensus first within our various stakeholders, whether it is manufacturing or engineering and quality assurance, to see that we are convinced that this technology is really transformative in nature and not a one-off automation which some organizations are looking for. So probably I think all these efforts can really bring a lot of value to the organizations. Even if you're operating from legacy systems, it can help you in good diagnosis and make sure that, to ensure that I need to prepare myself for future regulatory requirements. And as I was saying, that you have already have an annex one where a contamination control strategy needs to be worked out for everything, including your environmental monitoring, including your water systems. How do you assure that? So, do you have a sufficient data for that? I would say and recommend that more data needs to be generated at, especially first as offline mode in the legacy systems, and before validation and going to an online system. It can really bring a large change in the way we are looking at maintaining some of the water systems across the world.
So, as we close today's session, we express our heartfelt gratitude to our panelists for their time and for sharing such insightful perspectives. A big thank you to our attendees as well. We look forward to welcoming you again at our future events that focus on smart, data driven water operations. You may also join our PH2O Pure Water Technology discussion group on LinkedIn to be a part of some interesting conversations about clean utilities and related processes. Have a wonderful day, everyone.